Hi, class, and welcome to the Chapter 4 podcast. This is on the tour of a cell. I wanted to start with this picture right here. When I learned about cells, and I'm sure the last time you saw it was maybe in 7th grade, you had this picture of a cell with maybe a cell membrane around the outside, a nucleus, and that kind of an understanding of how cells work is pretty limited, and it also doesn't really get to how fascinating the cells are. And so I want to start with this. This is actually a die that shows you the cytoskeleton of a cell. And so that invisible part when we draw a cell, that clear, what we sometimes refer to as the cytoplasm, is really not so empty. In fact, it's this lattice of all these different microtubules and microfilaments, and they kind of work like monorails where we can move material around in the cell. I know we saw that video on the inner life of the cell, and so I want you to get this idea that cell is really exciting and really complex. And then the other thing I want to say right away is that it's hard to learn much about the cell and the way it operates unless you kind of get the terminology out of the way. And so make sure at the beginning of this chapter that you learn what is an endoplasmic reticulum, what's a Golgi apparatus, what is a mitochondria. So you have to kind of add those terms to your vocab. And if you don't know what they all mean, then you better figure that out right away. So the first slide I want to show you is kind of the discussion about why cells are small. In other words, when we look at all different types of organisms, we find that the cells that make them up, or this basic unit of life, is really small or tiny. In other words, it's invisible to us, and that's one of the reasons it took us a while to figure it out. When I mean we, I mean scientists. So why is it small? Well, this diagram tries to get at that. In other words, if we have this block, its total surface area is going to increase if we chop it up into a bunch of little blocks. So let me give you a better example of how that works. This is a Kleenex box. And so imagine I were to produce Kleenex boxes, but instead of folding all the Kleenex on the inside, I were to just make one Kleenex. And so when you wanted to blow your nose, you would blow your nose, or you would wipe it, or you would use it to clean up. And you'd find that pretty soon it's really gross. In other words, you would have used all the surfaces of the Kleenex box, and so you wouldn't sell a lot of Kleenex if you just had one big brick. And so instead, we fold a bunch of Kleenex on the inside, and so then we can use them over and over again. We've increased the surface area, and so that's how cells work as well. Now, you don't blow your nose on cells, but what do? You do chemical reactions along the edges of those. In other words, if you were to wait for oxygen to move all the way from the outside of this box or the outside of the cell to the inside through diffusion, you'd be waiting a long time. And so cells are small to increase surface area. In other words, we can't see it. They're that small. And so this diagram here shows what we can see with the unaided eye. But until the development of the light microscope, and this was developed by Anton van Leeuwenhoek, is the first microscope, and we actually have a copy of one of these in our room. Until we started actually using microscopes, we weren't able to see these cells. And so this would be the microscope that we use in class. It's just a compound light microscope, but we can use electron microscopes as well to get down into this level, looking at actual proteins and lipids. So we break microscopes into two different types. We have what are called light microscopes that use a series of lenses to focus light. You can get about 1,000 times magnification, which is much less important than your resolution. In other words, we can get to the level of 2 microns discrimination. Now, so in this picture right here, this is actually the cross-section of a leaf, and you can see individual cells. This would be a light microscope. But to go closer in, we use an electron microscope, and that uses a series of magnets to focus electrons. If you want to experiment with this, do not try this at home, but if you hold a magnet near your TV screen, do not try this at home. It will change the colors of the TV screen, and the reason it does that, it's actually changing the path of those electrons, and that's how an electron microscope works. Why should you not try that at home is it will permanently change your TV, and so it'll always have a spot in it or the wrong color. Now, electron microscopes have two different types. We have transmission electron microscopes where we get like a flat image of that. So this would be like a essentially cross-section of mitochondria. And then we can use a scanning electron microscope where you get a three-dimensional image. What's the good part of an electron microscope? We get much greater magnification and a much smaller resolution. What's the bad part? We can't look at living material. In other words, we have to coat it lots of times with a thin coat of gold before we can actually image it. And so they're dead by the time we look at them. Okay, cells come in two different types. We've got prokaryotic and eukaryotic. We'll go into the specifics of eukaryotic cells in just a second. But before we do that, I want to talk about prokaryotic cells. So these are bacteria. What are the parts of a prokaryotic cell? Well, they've got a cell membrane. Don't forget, all living cells have a cell membrane, or we sometimes refer to that as a plasma membrane, and that controls what gets in and what gets out. But they also have, prokaryotic cells have a cell wall around the outside. It's different in structure from the cell wall that you find in plants, but it serves the same purpose, and that is structural protection. Now they have DNA, of course, so they can make proteins and pass that information on, but it's not organized in a nucleus. It's just loose within the cell in an area called the nucleoid region. Another difference is that it's found in a loop. But then they have to have the other parts of the cell. In other words, to make proteins, they have to have ribosomes, and so they have ribosomes and they have cytosol on the inside as well. Now when we get to eukaryotic cells, then we find that compartmentalization occurs. In other words, we start to break down the specific jobs of that cell into little tiny organelles. In other words, in us, we have mitochondria to produce energy, breaking down sugar and eventually making ATP. But if it's a prokaryotic cell, they do that with their actual membrane. And so that serves that same purpose. In fact, we think that the mitochondria in us actually came from bacteria. So, without further ado, let's talk about the parts of a plant, the parts of a cell. The best way to think about it is we've got a code in here. That code is in the DNA, and so that's in the actual chromatin, which is DNA, and then these proteins called histones. Those are organized loose within the nuclei, and remember the DNA never leaves. When we eventually want to make something, and in a cell, that's, in other words, when we want to make a protein or some other macromolecule, the message is sent out from here, and so we make messenger RNA. That'll move out into the cell, where it's going to hit ribosomes. Now those ribosomes can be loose within the cell, or they can be attached to something called the endoplasmic reticulum. Endoplasmic reticulum is where we make proteins that are eventually going to go somewhere else. We also have smooth ER, and the function of that is lots of times to detoxify drugs. Smooth ER does that, but that's essentially ER that doesn't have ribosomes yet. So where do those proteins go that are produced by that rough endoplasmic reticulum? They then go to the Golgi apparatus. Think of that kind of like the cellular UPS, where these products come in, proteins, and then we package them to move either outside of the cell or somewhere else in the cell. They're carried by these vesicles to the Golgi apparatus, where they either leave the cell or are used within the cell. Now this is a cell, this is a plant cell, and so they're going to have these chloroplasts. We'll get more into the details of that when we talk about photosynthesis, but they have these membranes that absorb light, thylakoid membranes, and then they use that energy of the light to make sugars. That sugar is then moved within the cell to the mitochondria, where it combines with oxygen to produce ATP, or energy, and so that's what the cell uses. Of course, they have a plasma membrane around the outside, but since they're plant cells, they have this cell wall that's made of cellulose, and so it protects that cell. Now, plant cells also have what are called plasmodesmata, and those are big holes that connect adjacent cells, and so they can move large amounts of material between them. What am I missing in here? Some of the big ones. Oh yeah, the cytoskeleton is actually centered around the centrosome, but we have all these microtubules, which are big, and microfilaments, which are small, and those kind of form this cytoskeleton on the inside. Some of the things not in plant cells but are in animal cells would be like lysosomes. They don't have centrioles, and then flagella aren't found in plants, but they're found in certain reproductive structures in plants, just a few. Let's go to the animals then. If we look at an animal cell and how is that different from a plant cell, well, we can see the flagellum here is an example of something we'd find, or these villi on the inside. But the big thing that's missing, or the big couple of things that are missing, would be no cell wall. So we don't have a cell wall around the outside, and that's why humans are somewhat squishy compared to a tree. They also lack the chloroplasts, and so we can't make that sugar. We still have the mitochondria because we need to break down that sugar, but we lack that. And then also we lack this huge vacuole. The vacuole in plants, I should have mentioned that before, gives them structure, and so they can absorb and hold water inside it, and then it makes them turgid, or allows them to maintain their shape. And that's why if you don't water your plants, they'll wilt. Let's see if I'm missing anything else. Lysosomes are another one that they have. Lysosomes will actually carry digestive materials, and so we can break down organelles when they aren't operating correctly. Nuclear membrane is maybe one that I didn't mention, and again, that separates the nucleus from the cytosol, and that protects the DNA on the inside. And so that's kind of our quick survey of the cell, but you really have to dig in a little bit and figure out what all these organelles do on your own. I hope that helps.